Welcome back to the deep dive. Today, we're really digging into some critical research, especially if you're involved in ecology or managing natural resources.
That's right. We're looking at a paper titled, When Are Environmental DNA Early Detections of Invasive Species Actionable? The lead author is Adam J. Sepulveda.
In this deep dive, it's really focused on a practical problem. Managers get this cutting-edge data, this eDNA result, but it's uncertain. What do they actually do?
It's a huge question. The economic side alone is pretty mind-blowing. Aquatic invasive species, AIS, they've cost the global economy, what, over $345 billion since 1960?
Whoa, $345 billion.
And it's not slowing down. That cost is increasing exponentially. So the whole game plan relies on early detection rapid response, EDRR.
EDRR, right. That's the key. You've got to find the invader and react while the population is small, manageable.
And that's where environmental DNA, eDNA, comes in. It seems like the perfect tool for EDRR.
But it's so sensitive.
Exactly. Incredibly sensitive. You can detect tiny traces of an organism's DNA in the water much, much earlier than you'd likely spot the actual creature.
Often cheaper, too, right? Compared to sending crews out for physical sampling.
Often, yes. Lower cost, earlier detection. Sounds like a win-win.
Yeah.
The ultimate early warning system.
Okay, but here's the catch, the paradox we mentioned. That sensitivity, it brings uncertainty.
A positive eDNA test doesn't automatically mean you have a living, breathing, reproducing population there.
And it could just be DNA fragments washed in from upstream.
Yeah,
Or maybe contamination from, say, gear or a boat.
precisely. And that puts resource managers in a really tough spot, a real decision paradox.
So do you launch a massive, expensive response based on what might be a false alarm? You could waste resources, restrict lake access, stir up public concern, all for nothing.
Or do you hold back? Do you wait until you have boots on the ground, visual confirmation?
But if you wait, and it is real, the invader gets a foothold. It establishes, maybe spreads, and suddenly control is way harder, maybe even impossible. The EDRR window slams shut.
So that's what this paper tackles head on. How do you bridge that gap? How do you move from an uncertain eDNA signal to a sensible management action in a way that's transparent and defensible?
And they use a specific framework to do this.
Yes, structured decision-making, or SDM. It's a tool designed exactly for these kinds of messy problems where you've got uncertainty, multiple goals, and different stakeholders who might want different things.
Okay, STM. How does that work, basically?
Well, it uses a process often called PRO-OACT. That stands for defining the problem, clarifying the objectives, identifying alternatives, evaluating the consequences, and then analyzing the trade-offs.
The objectives part sounds key. It's not just about find the invader.
Absolutely not. SDM really forces you to involve stakeholders and get crystal clear on the fundamental objectives. What do people actually value about this resource?
So it goes beyond just the ecology.
Oh yeah, you're looking at impacts on local economies, livelihoods, maybe cultural values, political realities, property values, recreation. It's holistic. You define what truly matters first.
Because if you don't, you might end up, I don't know, perfectly controlling the invader, but destroying the local tourism economy in the process.
Exactly. You might optimize for a means objective, like confirm eDNA, instead of the fundamental one, like maintain water quality for drinking and recreation.
But doesn't this SDM process doesn't take a lot of time? You mentioned EDRR needs to be rapid.
That's a really important point, and the paper acknowledges it. The previous study they reference, which also used SDM for eDNA, took months to complete the process.
Months? That doesn't sound very rapid response.
Well, and this is crucial. The paper argues the real power of SDM isn't necessarily as an emergency tool after you get a positive hit.
Ah, okay. So when do you use it?
The ideal time is before detection. You do the SDM work early as part of your eDNA monitoring planning.
Pre-planning, like a fire drill for invasive species?
Kinda, yeah. You get all the stakeholders together, you define the objectives, evaluate the potential actions, their trade-offs in advance. So when that positive eDNA result does come in, boom, the plan is already there, vetted, and agreed upon.
Turns months of potential argument into immediate planned action. That makes a lot of sense. Do the hard thinking when you have the luxury of time.
Exactly. Now, to see how this works in practice, the researchers applied the SDM framework to two very different real-world scenarios.
A tale of two water bodies, as they put it.
Right. The first was Jordanelle Reservoir in Utah. They were looking at Dreissenid mussels, zebra, and quagga mussels.
Oh, those are the really bad ones. Infamous.
Truly among the worst globally. They clog pipes, cover surfaces, change entire food webs. They can impact the whole water column, the bottom, everywhere. Huge potential impact. high risk.
Okay, so that's case one. High-impact invader, what's the contrast?
Case 2, their main focus for this paper was Sebago Lake in Maine, a very different system and a different invader.
Sebago Lake. Isn't that a major drinking water source?
Huge. Supplies drinking water to over 15% of Maine's population, and it's known for incredibly high water quality, so high it has a federal filtration exception. That's a big deal financially and ecologically. It's also large and deep, up to 96 meters.
Okay, pristine, important lake. What was the invader there?
An invasive plant, Najas minor. Common name is European naiad. It's a freshwater macrophyte, meaning a rooted plant.
So not like the mussels that can cover everything.
Right. Najas minor is basically restricted to the littoral zone, the shallower, near-shore areas where sunlight reaches the bottom so it can root and grow.
In a big, deep lake like Sebago, that automatically limits its potential reach compared to the mussels.
Exactly. It can form dense mats in those shallow areas, which isn't good, but it's unlikely to have the same kind of system-wide catastrophic impact as draconids in that specific deep lake context.
Are there other factors about this plant?
Yeah, it's an annual, reproduces by seeds, and its stems are brittle, so physical removal can be tricky. You might just spread fragments and seeds. There are cost-effective controls like herbicides for when you find it, but the key thing is its impact is more geographically contained in a lake like Sebago.
So the main working group used the SDM process for Najas minor in Sebago. What objectives did they prioritize?
They came up with 7 fundamental objectives. Top of the list, absolutely critical, was protecting Sebago Lake's water quality.
Because of the drinking water and that filtration exemption.
Precisely. They measured this using the Trophic State Index, or TSI. If that index slips, the exemption could be lost, costing millions. So TSI was a direct proxy for massive financial risk.
Makes sense. What else?
Minimizing any additional costs related to drinking water treatment. Also, crucially, minimizing the spread of any AIS beyond Sebago Lake into connected waters. Maine really values the pristine nature of its other lakes and rivers.
So containment was a big priority in itself.
A huge one. Then also protecting the lake's ecosystem structure, things like the native landlocked Atlantic salmon population, ensuring the response plan was affordable and sustainable long-term. And, of course, protecting economic value, property values, tourism, and maximizing recreational access.
That's a lot to balance. Water quality, salmon, costs, tourism, stopping spread.
It is, and that's why SDM is helpful. It forces you to articulate all those competing values and then evaluate how different actions impact all of them, not just one.
Okay, so how did they model this? What were the potential actions they looked at?
They set a 10-year time frame for their analysis. They modeled the probability of lake being in one of 3 states. D, just detectable eDNA. I, isolated infestation, which they defined as less than 20 acres. Or W, widespread, meaning more than 20 acres.
And they looked at different management alternatives.
Yep. Ranging from the baseline, the status quo, which involved existing containment efforts like boat inspections and education, all the way up to more aggressive options. Things like restricting surface use in certain areas or using chemical controls, like herbicides.
And they modeled how each alternative performed against those 7 objectives over the 10 years.
Correct. Using things like expert opinion and ecological models to estimate probabilities and consequences, they calculated a cumulative performance score for each alternative.
All right, so the big reveal. What did they find was the best approach if they got an eDNA hit for Najas minor in Sebago?
This is where it gets really interesting and, frankly, quite unexpected. The optimal strategy, the one with the best overall performance score immediately after detecting eDNA, was to basically do nothing new.
Nothing.
Well, specifically, Alternative 1A, maintain the current status quo containment efforts and take no additional management action based only on the eDNA detection.
Wow, okay. But what if they later found the plant, like saw it visually?
Even then, the modeling showed that even if the plant was subsequently confirmed using non-molecular methods, the best course of action, Alternative 2A, was still to return to the status quo.
So find the DNA, do nothing extra, find the actual plant, still do nothing extra beyond the existing boat checks.
That's what the model indicated was optimal for Sebago Lake and Najas Minor, based on their objectives and trade-offs. The alternatives that included active control, like opicide treatment, Alternative 2-pen, consistently ranked lowest in overall performance.
That feels completely backwards. Isn't the whole point of EDRR to jump on it immediately? You find it, you hit it hard.
It feels counterintuitive, I agree. But it makes sense when you unpack the why, based on the SDM trade-offs.
Okay, explain the why. Why did status quo win?
Because the existing status quo actions, primarily, Those boat inspections and outreach programs aimed at preventing introductions were already quite effective and cost-efficient at addressing one of the highest priority objectives, minimizing spread beyond Sebago Lake.
Ah, so the prevention piece was already strong and working on the biggest risk factor for other lake.
Exactly. Those existing measures provided good value for that crucial objective. Now, compare that to the alternatives involving active control or use restrictions.
Those would be more expensive.
Much more expensive, yes. And they would have directly negatively impacted other high-priority objectives, like maximizing recreational access and protecting economic value tied to tourism and property values.
Right. Restricting boating or using chemicals, people wouldn't like that, and it costs money.
And crucially, the potential negative impact of Najas Minor itself within Sebago Lake was deemed relatively low and localized, primarily affecting those shallow littoral zones. It wasn't seen as an existential threat to the lake's overall health or, importantly, its water quality and that federal siltation exemption.
So the cure was potentially worse than the disease in this specific context.
That's a good way to put it. The significant costs and negative social economic impacts of aggressive action outweighed the projected benefits of controlling a plant whose impact was limited in that large, deep lake system. The trade-off analysis favored accepting a low, localized impact over high-cost, high-disruption interventions.
Okay, that makes sense when you lay it out like that. Now, contrast this with the Jordanelle mussel case.
It's night and day, really. For Jordanelle, with the high-impact graysinid mussels, the eDNA detection was considered highly actionable.
Why the difference?
Two main reasons highlighted by the comparison. First, the potential impact of mussels was vastly greater system-wide, ecologically and economically devastating. Second, Jordanelle didn't have the same level of robust pre-existing containment measures in place that Sebago did.
So high potential damage and less baseline protection already happening.
Correct. So for Jordanelle, the optimal path identified through their earlier SDM work was immediate follow-up with non-molecular methods to confirm, quickly followed by implementing mandatory watercraft inspections and likely decontamination. The eDNA signal demanded an escalated response.
Whereas for Sebago, with the lower impact plant and strong existing containment, the eDNA signal was not actionable in terms of triggering new aggressive measures.
Precisely. The best action was to just continue the effective, cost-efficient status quo containment.
It's fascinating how different the conclusions are based entirely on the context.
It really highlights that there's no one-size-fits-all answer for eDNA results. But there is a common thread that emerged, despite the different outcomes.
What's that?
Containment actions. In both studies, strategies focused on containment preventing spread to other water bodies either were the optimal choice, Sebago, or performed very well and were a key part of the recommended escalation, Jordanelle.
So that reflects a universal high value placed on prevention, on protecting currently uninvaded waters.
Absolutely. It's almost always cheaper and more effective to prevent spread than to control an established population. Those boat inspection programs, even if simple, seem to provide a lot of bang for the buck in the eyes of managers and stakeholders.
There was also a policy difference, wasn't there?
Yes, another key contextual factor. In Maine, there's a specific statute they cite, Title 38, Section 1864, that essentially requires visual confirmation of an invasive plant before certain intensive control actions, like widespread herbicide use, can be legally undertaken.
So the Sebago manager's hands were tied to some extent by law, reinforcing the wait for confirmation approach.
Correct. Whereas the decision makers for Jordanelle apparently had the policy latitude to consider more aggressive actions based solely on the eDNA detection, pending confirmation. Policy shapes the possible response pathways.
Okay, so putting it all together, the paper moved towards a general framework, but the big takeaway is it depends.
Yes, in a way, the actionability of an eDNA detection is fundamentally context-dependent. You have to consider the specific habitat, the invasive species' potential impact of that habitat, and the existing management and policy landscape.
But they did offer some general rules of thumb, some heuristics based on these two cases.
We did. Sort of guiding principles for managers trying to make this call.
What are they?
Okay, heuristic one, based on the Sebago case. eDNA is likely not actionable for triggering new aggressive interventions if effective containment is already in place, the invader's potential systemic impact is minimal, and policy requires confirmation before major action.
Makes sense. And the second one?
Heuristic 2, from the Jordanelle case. eDNA is likely actionable if effective containment is not currently in place. The invader has considerable potential for widespread impact, and policy allows for rapid response based on detection.
So two ends of a spectrum. Low-impact cloud containment versus high-impact cloud containment.
Right, and that points back to the value of doing the SDM work beforehand, especially for situations that fall between those two extremes.
What kind of situation would that be?
Imagine you have good containment already in place, like Sebago, but the potential invader has KGH impact potential, like the mussels. That's a really tricky scenario.
Yeah, the existing containment might not be enough, but launching a huge response is still costly.
Exactly. Those are the cases likely to benefit most from a detailed pre-detection SDM exercise to figure out the optimal triggers and responses before the crisis hits.
Now, the study did have some limitations they acknowledged, things they didn't look at.
Yes. Importantly, they focused on the implications of positive detections. They didn't explicitly model the consequences of false negatives.
Meaning, the invader is there, but the eDNA test misses it.
Right. That's a whole other layer of risk. If you get a negative result, assume you're clear and relax efforts. But the invader is actually present and establishing. You've lost that crucial early detection window entirely.
So the uncertainty cuts both ways, acting on a false positive, or failing to act because of a false negative.
It's complex. They also noted the need for more case studies, particularly, as we mentioned, invaders with known significant ecosystem impacts, but where there are also proven cost-effective control tools available.
To really understand when active control, like chemical or physical removal, becomes the better option compared to just containment.
Exactly. To find that tipping point in the cost-benefit analysis.
So, wrapping up, the core message seems to be that eBNA is incredibly powerful, a revolutionary monitoring tool.
But it's still just a tool. And like any powerful tool, using it without a clear plan and understanding of the context which SDM provides can lead to trouble.
You risk making really costly mistakes, or sparking major public controversy, potentially diverting resources from where they're most needed.
The SDM process, even though it takes time up front, builds that confidence. It ensures that the actions taken are truly aimed at the fundamental goals protecting water quality, stopping spread, preserving economic value, not just reacting to an intermediate signal like a positive DNA test.
It shifts the focus from the means, the detection method, to the ends, the actual desired outcomes for the ecosystem and the people who rely on it.
Well put.
So the final thought we want to leave you with is this. Think about your own field, your own work. Whether it's managing resources, running projects, making business decisions. Where are crucial choices currently being made reactively, perhaps in response to incomplete data or the latest fire? And how might the outcomes improve if you paused before the next crisis or detection hits to clearly define the fundamental objectives and trade-offs involved?